Hello, everyone. Welcome to today's live webcast, Navigating the Analytical Development Challenges for Bioprocess Residuals and Impurities. I'm Rita Peters, Editorial Director of Pharmaceutical Technology and Biopharm International. Today's educational webcast is brought to you by Eurofins Lancaster Laboratories. Founded in 1961, Eurofins Lancaster Laboratories provides analytical testing for nearly all stages of the drug development process and supports all functional areas of biopharmaceutical manufacturing, including analytical development, microbiology, process validation, and quality control. As a member of Eurofins Scientific's Biopharma Product Testing Group, the largest network of harmonized biopharmaceutical GMP product testing laboratories worldwide, Eurofins Lancaster Laboratories provides comprehensive laboratory services for the world's largest pharmaceutical and biopharmaceutical companies. The company's service offerings are fully comprehensive and include testing of drug substance, final product, intermediates, and starting materials for both small and large molecule drug products. Eurofins Lancaster Laboratories also offers clients the flexibility to choose from 5 service models to meet specific project needs, including the award-winning professional scientific staffing insourcing solution, and also provides secure 24-hour data access via its online data portal, labaccess.com. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the red Q&A widget at the bottom of the presentation window. We'll try to answer as many questions as we can following the presentations. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark Help widget in the dock at the bottom of your presentation window. I'm pleased to be joined today by an expert panel to discuss this topic. Sheila McGill, a senior consultant with Bioprocess Technology Consultants, has over 20 years of experience in quality and analytical method development for biologics, peptides, and small molecules. Her expertise includes quality assurance, protein and peptide biochemistry, and analytical development. She was formerly Senior Manager of Analytical Development and Quality Control at BioMeasure, Inc., and previously held positions at Warata Pharma, Alkermes, Bion, and HHMI at Massachusetts General Hospital. Dr. McGill has implemented quality systems and has managed external analytical and QC activities for multiple biopharmaceutical products. She holds a PhD in biochemistry from the University of Minnesota. Dr. McGill will provide an overview of analytical development challenges for bioprocess residuals and impurities, including regulatory expectations. Daniel E. Peckman has been with Eurofins Lancaster Laboratories since 2009 and applies 16 years of experience in the biopharmaceutical manufacturing, quality control, and testing industry to his role as manager of Eurofins Lancaster Laboratories Biochemistry Department. His expertise with bioseparations development, process residual testing, and biomanufacturing lends insight into method development for bioprocess residuals and impurities testing. Mr. Peckman earned a Bachelor of Science degree from Messiah College and a Master's of Science degree from Towson University. He'll discuss detection types and method development for bioprocess residual analysis. Dr. Weihong Wang, Technology Development Manager for Eurofins Lancaster Laboratories Molecular and Cell Biology Group, oversees assay development and validation projects, and serves as a subject matter expert and direct technical contact to assist clients with testing needs. Dr. Wang has 12 years of experience developing and characterizing biopharmaceutical products and has extensive experience with ELISA and qPCR-based residual assays. She previously worked for Wyeth Biopharma, which is currently part of Pfizer, and Johnson & Johnson companies, and participates in USP and FDA task forces. She earned a PhD in cell and molecular biology from Brandeis University and completed postdoctoral research at Harvard Medical School. She will discuss molecular biology methods and immunoassays for quantitation of bioprocess residuals. Thank you all for joining us. And Dr. McGill, please start us off.
Good day, everyone. As Rita has said, I've been asked to give a brief introduction to sort of put the topic in perspective and, more importantly, to talk about what the regulatory expectations are. First and foremost, let's discuss the difference between a residual and an impurity. All residuals are impurities, but not all impurities are residuals. And by that, we mean that impurities are product-related materials which may be either a component or a non-component of the drug substance, but related to that, or may in fact be an excipient in the drug product, a material that comes in with that but is not itself the excipient. These may be aggregates or clipped forms or other degraded materials of the product itself. Impurities are usually classified into the following categories, either organic, inorganic, or residual solvents. And the ICH has put out a number of guidances which have been accepted by the FDA as well as the EU and Japan Medical for residual solvents, and those can be obtained online. Organic impurities usually arise during the manufacturing process or possibly during the storage of the drug substance or the drug product. They're identified or unidentified. That is to say, we don't always know what some of those materials are, and they may be volatile or non-volatile. Some of the materials which would be considered an organic impurity would be remnants of starting materials or byproducts, intermediates which are not fully processed but still remain within the drug substance or the drug product, as well as degradation products of the active ingredient. Additionally, there may be residuals of the reagents or ligands that were catalysts used during the manufacturing. Inorganic impurities almost all result from the manufacturing process, and they're usually understood and identified. And these can include reagents as well as heavy metals or residuals, inorganic salts, and some filtering processing materials. It's a truism that if you don't know where you're going, you will never get there. And this actually represents one of the biggest hurdles in measuring residuals in early development. During early development, you don't really have a good indication of all of the quality attributes. You don't understand what the potential residuals are, in part because the process itself is not yet fully finalized. And the degradation pathway of the active ingredient may not be understood. Complicating this is the fact that the methods that one applies, some of which are, if not strictly platform methods, that is methods that a company would have that it uses routinely, they are methods that are general purpose within a biochemistry or protein chemistry laboratory, such as SDS page or size exclusion chromatography. But you haven't demonstrated that those methods are suitable due to limitations which may be due to the sample or the sample matrix itself. Now, why is it important to know what the impurity levels are? Well, obviously, it's a regulatory requirement, and so it cannot be ignored. But equally, if not more important, are the risks to both the patient and the operator. For the patient, impurities may be toxic. Some impurities, such as aggregates, are known to be immunogenic, which can, in fact, cause antibodies to be produced to the active form. And these, in turn, can minimize the effectiveness of the drug. Additionally, some impurities may compete with the drug. If an antibody product is working by binding to a particular receptor, certain impurities may also bind to that receptor interfering with the proper binding of the drug of interest. The operator risks usually are exposure to toxic materials which are either used during or created during the manufacturing process. And there are, of course, product risks, such as a reduction in the effectiveness or, equally of concern, an increase in the degradation rate of the product itself. Now, some impurities are relatively easy to measure, such as a residual detergent. In that case, the impurity doesn't look like the product and is much more easy to either separate from the product or to actually measure in the presence of the product. Other impurities, such as residual host cell proteins, while they do not resemble the product, are still more difficult to measure because they are so heterogeneous. And more difficult still are those materials which look very much like the product, such as charge variants. So, if you will, the closer in structure to the product, the more difficult it is to measure most impurities. Added on top of that is, as one moves through development from preclinical to commercial, the acceptable level of certain impurities is decreased. Now, that has some exceptions, most particularly the safety materials, such as endotoxin, which has an absolute limit. But it is, if you will, a truism that the closer one gets to commercial, the greater the need to reduce these impurities, which also means the more sensitive the methods for measuring them must be. Some process residuals in general differ from the structure of the product, as I've already mentioned, and are more readily separated. But the regulatory concerns are based either on known or believed impact on patients. Now, some process residuals are cell substrate related, cellular debris and cells, which are usually very readily separated early in the process. But residual DNA from the host, as well as host cell proteins, may in fact still be present when the final purified material is delivered. There may also be materials coming in from the culture media, such as antifoam or inducers or antibiotics, as well as endotoxin, which may enter not only during the cell culture, but at a number of points throughout the development and purification as it comes in with some of the reagents. And then, of course, purification reagents and materials. One of the biggest concerns, because of the increased number of antibody products being developed, is protein A. Protein A is one of the most common first steps for purification of an antibody, but the protein itself often is released from the resin during processing. And so it is important to measure that residual present at the end. As mentioned, endotoxin has to be measured. And one of the areas that often doesn't get as much attention are leachables out of plastics that are used either during the process or to store the drug substance or drug product itself. Product-related residuals are more complicated. They resemble the product, making them much more difficult to separate and much more difficult to measure in the presence of the product. On top of which, some product-related residuals may themselves have activity, which may be an agonist or an antagonist or a pure competition to the product. At each step of the purification, you need to balance removal of these product-related residuals against product yield. Sometimes that's easy to do because it is known that at a later step this type of product-related impurity or residual will be more readily removed. But in some cases, that isn't the case. Some kinds of product-related residuals, as mentioned, are degraded or aggregated forms, and the presence of aggregated forms is of a significant interest to regulators because of the known or believed impact in raising antibodies to the active product. Development of suitable methods for these materials is often hampered because one cannot create all the possible forms and then evaluate those in the method. Now, what are the regulatory expectations? Well, specifications have to be set for residuals and impurities. And as I've already mentioned, during development, you need to demonstrate increasing control over these materials. The analytical methods have to be suitable. They have to be developed early and qualified and validated late. Now, qualification of an assay is one of those words that doesn't have a real definition. The working definition for qualification is a demonstration that the analytical method is suitable for its intended purpose. That also reflects out to those methods which are used to evaluate the stability of the product. You have to demonstrate that a stability method, in fact, is capable of demonstrating if the material were unstable or an increased degradation took place. One can do a retrospective analysis, that is, measuring the residual at the end of the process. And this is frequently what is done early in development, and it is acceptable early in development. But regulators will expect that a process that monitors the residual multiple points during the process to demonstrate that it is removed or cleared is what is going to be presented. And certainly at the time of filing of a BLA, clearance studies for certain impurities will be expected. Now, I haven't talked about viral clearance studies, which are an absolute requirement and are covered by a number of guidances. So I'm not really going to talk about that now, other than to say early in a process, one needs to demonstrate viral clearance for 2 model viruses. Late in the process, before BLA filing, 4 model viruses must be shown to be cleared or inactivated by the process. To put some numbers around the regulatory expectation. Early in phase 1, host cell protein may actually be measured by a commercial kit, and for a number of the common cell lines used to produce biopharmaceuticals, these kits do exist. The levels that need to be demonstrated are in the microgram per gram of active protein. It may be that a commercial kit will be suitable beyond phase 1 or phase 2, but in that case, one would have to demonstrate the suitability of that kit. Residual DNA at phase 1 is usually acceptable if it is less than about 50 nanograms per dose. Endotoxin, which is a compendial safety issue, has an absolute upper limit of 50 endotoxin units per kilogram per dose. Now, that is an upper limit, and the expectation is during development that the process will reduce endotoxin below those levels, and specifications will be expected to be tightened even for endotoxin during development. By time a product is about to be filed for commercial, host cell protein is expected to be measured by a product-specific method. As I mentioned just before, a commercial kit may be suitable, but one would have to demonstrate that the multiple kinds of host cell protein produced by the cell line actually being used are measured to the greatest extent, and that's usually greater than 90% of them that might be present by the kit. And at that point, expectation is that host cell protein will be reduced to about 100 ppm or less. DNA should be at this point at 10 nanograms per dose or less. Now, I know that historically the number that was used was less than or equal to 10 picograms per dose. That was set very early in the history of recombinant protein and was based on estimates and guesstimates. One of the things that I should point out is even though the level of DNA is kept low, there is an expectation as well that the size of the DNA strands are kept low. The larger the size of a DNA strand, the greater the expectation that it may incorporate oncogenes, and that is the biggest concern for residual DNA. As I mentioned, endotoxin has an upper limit of 50 EUs per kilogram per dose, but if during development it is uniformly shown that the level of endotoxin can be reduced significantly below that, then that is what the expectation on the part of the regulators will be, and the specification will be expected to be set at what is seen historically. Thank you very much. And I'd like to now introduce the second speaker, Dan Peckman of Eurofin's Lancaster Lab, who will talk about some more specific examples for you.
Good afternoon, everyone. My name is Daniel Peckman, and I will be talking about analytical development challenges for bioprocess residuals. This presentation will basically be an overview of the thought process and some of the analytical techniques that go into achieving residual tracking and allowing us to show clearance through the methodology. So really some of the things we need to take into consideration, we have a wide variety of additives going into the bioprocess. We need to track their clearance and aid in that establishment throughout the process characterization. Primarily, we'll be talking today about HPLC-driven clearance techniques. These include a wide variety of detection techniques, and we'll also be touching on some advanced techniques such as LCMS-MS and electrochemical detection that can help us drive down the level of detection even lower than traditional methods. Just a quick overview of the types of things that we may be interested in tracking. Many of the additives that go into bioprocess will be introduced throughout the processing of the biologic. There could be fermentation additives, there may be reducers, chaotropes, there may be different types of antibiotics, either very early in the process or used as plasmid retention items. We also see a lot of downstream additions to aid in the chromatography. And so there's really a wide variety of things that may be put into the process that we need to track and then show clearance for. Really, the big challenges that we face with developing and using these types of methods is the variety and the complexity of the matrix. The bioprocess is extremely complex, and we may be looking at many different matrix types across a single process, especially if there are many chromatography steps. So the target residual that we're looking at may be only one of the components that can interfere or bind to the protein and cause issues. So we first want to look at removal of protein. That may be necessary. We may be actually adding other additives to the sample to allow us to release the residual from the protein. So these are all things we have to take into consideration. We also need to make the decision of how sensitive the method really needs to be. Can we live with a part per million approach? Is the residual to the point that we really need a part per billion level of sensitivity? That's also something we need to take into consideration, because that's really going to drive our decision-making process with what type of chromatography system and detection technique we use. So there's lots of specialized instrumentation that we could use, but we need to make some choices up front. We need to look at whether the level of sensitivity we need is going to add in some other challenges. In other words, if the detection system we choose is going to also add layers of complexity when it comes to increased noise, background signal, things like that. We also have problems that come along with increased sensitivity, which are, system cleanliness needs to be a paramount concern, laboratory cleanliness, as well as really doing everything we can to avoid any type of cross-contamination during the process of testing. So the common question is always, how low can we go with the detection? And Sheila gave some guidelines, some outlines, but usually early on in the process, the decision needs to be made, where are we going to target the residual sensitivity? And that's going to dictate what type of method we choose. If we're looking at ppm levels, we may use a traditional detection technique that has a level of reproducibility between 1 and 10 ppm. If we need to go lower than that, we may need to go to a more advanced system to achieve lower detection limits. One of the most common detection types we use is a charged aerosol detector. This is a universal detector. It has some benefits in that it's fairly cost-effective. There's not a lot of time for development as long as you can adjust your chromatography accordingly to the limits of the detector. It's an excellent limit test and screening technique, and it can set you up for transitioning to mass spec in the future if necessary. It can be made a quantitative method, but charged aerosol detectors from instrument to instrument can give different responses. So it works best as a screening tool and usually does not perform as well as a QC tool. The mass spectrometer is an excellent tool for very high sensitivity, and we use that considerably on any item that we can ionize. So coupled with an LC separation, the process by which we would perform this would give us an excellent quantitative analysis. If you can see your standards, ionize your standards, tune for your standards, you can get excellent sensitivity on something like an LC-MS, where you can actually not rely as much on your separation as you do the actual power of the instrument for identification and quantification. Some things you have to consider are your sample matrix and any components that may suppress ionization, such as salts, sugars, polysorbates. You're you're going to need to consider those in the sample prep so that you can get a reliable response from the LC-MS. Just a few general detectors that may be employed but will most likely be very selective in our choices based on the nature of the residual. Refractive index can be used where appropriate, but you don't have options for gradients, so your chromatography will be limited, and it's not as robust as, say, a UV detector. It can be very good for limits and screening, but it's not necessarily appropriate for all techniques. Ultraviolet detector is very cost effective. It's readily available. However, a lot of the residuals we're looking at have no profile, so that's really not something that may be a benefit. If you cannot remove your protein as well, UV detection is going to be problematic. One very focused technique that we have employed is the electrochemical detector. This can be tuned to give excellent sensitivity comparable to an LC-MS, which would be in the low ppb level. It is a bit time-consuming to develop and can be time-consuming to set up. It's an excellent QC detector, but you really need to be careful with your matrix effect, as you have samples with a variety of buffers will dramatically impact the detection cell and will each need to be specifically developed to be able to be managed on the ECD detector. Some things to consider with methods development are column selection instrumentation. That really is going to drive your entire process. So you need to be careful about what columns you're going to select and whether or not they're really going to provide the type of selectivity that you need and the sensitivity that you need for the specific residual. You want to consider preparing standards at many different levels, but your priority needs to be at the low end. So if LOD and LOQ level standards are viable, then that gives you a pretty good sense that you're going to be successful with the chromatography. More things we want to consider are sample preparation. Typically in the biochemistry field, we're most interested in protein. However, whenever we're talking about biochemical residuals, we really have to consider removal of the protein as the most important part of the testing. Most of the chromatographic procedures are going to have problems if the protein remains and or the actual residual we're looking for is going to stick to or be interfered with by the protein. So we need to look at precipitation and filtration possibly, and also identify matrix components that could potentially interfere, such as sugars or polysorbates that may interfere with the actual precipitation of the protein. The other thing that's really critical is start thinking about how we're going to spike and recover your target from the samples in question. Many times residual testing will show almost no signal on your target of a sample, but that's really only as good as your spiking technique, and typically spike controls at multiple levels need to be assessed to verify that your technique is functioning consistently. Some of the ways that we could address the protein can go from basic cleanup steps like precipitation to more involved columns, and these types of things can limit your spike recovery. So we really need to be careful in the prep as to how many perturbations we're adding into the samples. Samples are spiked typically at the beginning of the step, and then we would go through the preparation to really mimic how a sample would arrive and what our preparation is going to do to that residual and whether we're going to see any loss. A general rule that we use is an average recovery of 70 to 130% of the theoretical is considered successful for a residual method, and that will vary greatly depending on the level of the residual that we're looking for. And relative standard deviations around 15% or less would give us a pretty strong indication that we have a fairly reproducible method. So what do we do if we're not meeting the criteria? Well, a lot of times we'll default to increasing dilution, but that's really going to hurt our sensitivity level that we can report. The other thing we need to do is determine whether or not our recovery is due to protein. We may have to look at different ways to actually remove the residual from the protein. We could reevaluate the methodology concerning the sample starting type and look at alternative detection and column choices. One question that comes up a lot is platform methodology, because in early phase and looking at a lot of samples at one time, it's always a good idea to have a platform approach. Sometimes this is possible, sometimes this is not. Typically, platform methodology will give you a good foundation for your separation, but you still have to deal with the different buffer and matrix components for each of the process steps. So in some cases, platform methodology can be a very useful tool up front, but you really have to pay attention to how much your matrix changes are going to affect your output of the method. And that's really where spike recoveries, checking levels of recovery at each matrix that changes needs to be part of the method. And if that is successful, then the chromatography can remain the same. I wanted to go over a typical residual impurity example that we would look at. The scope of this example is an antibiotic. A lot of times, like I mentioned before, these are introduced upstream, and we need to show and demonstrate subsequent process clearance, and not only throughout the process, but there will normally be a bulk drug substance assay shown to have eliminated the antibiotic throughout the process. So some of the challenges are we need to see a good acceptable recovery from the process matrices. So the mass balance of the process, if we know what we're putting in, we want to be able to show that clearance. Any interferences from other potential products in the process, we need to look at that. This antibiotic in question is kanamycin that we're going to look at, so we don't really have a good UV profile. And but the upside is we can use a traditional chromatographic approach to separate. Detection will just be the key. So in this case of kanamycin, we actually went ahead and spiked the sample with an internal standard, amikacin. We went through a protein precipitation followed by a molecular weight cutoff and analyzed the supernatant on a traditional ion pairing HPLC separation, and then went into detection by MSMS. Just a quick overview of the instrument parameters gives the setup of the triple quad. As you can see, the mobile phase A and B are typical reverse phase ion pairing with a C18 column and some transitions that we would use to actually isolate. The amikacin and kanamycin elute in a very similar manner, which is excellent in regards to the use of an internal standard, and are shown to be linear over a wide range, well into the low parts per billion level. So a discussion at this point would show that with MS-MS, reverse phase ion pairing gives a straightforward approach. It's not difficult to set up, and we can see levels quite low. One common challenge with very low level detection is the residual presence of the target remaining on the system. This is an ongoing challenge because the sensitivity of the triple quad is such that we really need to be very careful about standard preparation, not allowing enriched samples onto the system, and how the actual samples are prepared so to avoid cross-contamination. The real take-home out of dealing with this issue is we always need to have limit of detection standards prepared and run each time we analyze a set of samples. This gives us very good assurance not only that our blanks are clean, but we also can monitor response differences between instrumentation, which may be quite different depending on which system is being used. So the conclusions out of this really are that it's critical to have upfront information about the sample you're working on. Matrix and the process that the sample is coming from really needs to be understood if you're going to design an experiment for the detection of the residual present. I would say consider using limit tests early in development to trend your clearance and allow time to build up some data before you really dive into quantitative approaches, because they can be problematic before you're sure you know what you want to track. You want to keep the sample preparation as simple as possible to avoid additional steps that are going to give you recovery problems, and you want to keep throughput very high early on while you're figuring things out of your process. That concludes the portion of my presentation. Now I'd like to introduce Dr. Wei-Hong Wang. She will be our next presenter. She will be speaking on bioprocess residuals from a cell and molecular biology point of view.
Thanks, Dan, and thanks everyone again for attending our webinar. You've heard Dan talk about various biochemical methods for the detection of bioprocess residuals. My part of talk will be focusing on molecular biology methods as well as immunoassays. These methods actually have been proved to provide sensitive quantitation of certain types of bioprocess residuals, and most commonly these include host cell DNA, host cell protein, growth factors or cell culture additives such as human or bovine serum albumin, insulins, and transferrin, etc. There are also other process-related protein residuals oftentimes introduced through downstream purification, such as benzonase and protein A. Now, first I want to focus on the host cell DNA. There are quite a few different host cell DNA methods that have been used in the biopharm industry. However, not all methods are created equal. For example, the historical method has been the hybridization or the slot blot methods, and then subsequently there's methods that utilize immunological interactions between DNA and DNA binding proteins. These are, most common ones is the threshold methods that a lot of people have used. And more recently, there's non-specific DNA dye-based method such as picogram that was used. However, my talk will be focusing on qPCR because qPCR method has been proven to be the most commonly used technique for detecting host cell DNA. And it is of good reason because qPCR methods offers superior performance. This includes extreme sensitivity. Typically, qPCR methods can detect as low as picogram per ml DNA. And qPCR is also very specific and provide large dynamic range, oftentimes more than 5 logs of detection range. Another unique aspect of qPCR is that it is capable of detecting small DNA fragments, oftentimes down near or below 100 base pair. You've just heard Sheila talking about, the larger the DNA size is, the more concerned it is, because there are some, tumorigenicity concerns before. So being able to detect a small fragment of DNA is of a great advantage. And also the qPCR method has been reviewed and accepted by regulatory agents for many years now. For whole cell DNA, matrix effect, as in many other different residual testing, is of a concern. Protein products and other matrix components often interfere with qPCR detection. Therefore, DNA extraction sometimes may be necessary prior to qPCR reaction. Typically, the extraction procedure includes a proteinase K digestion to get rid of the protein components, and then subsequently DNA purification via either phenylchloroform, chemotropic agent methods, a silicon column kit, or magnetic beads-based kit. Sometimes when the matrix effect is more severe, buffer exchange or dilutions prior to sample extraction may be necessary. So for whole cell DNA, proper controls and appropriately assigned system suitability criteria is essential in assuring validity of the method. The most commonly used and crucial controls within the assay are, first, negative controls. These are either blank or assay buffer that are subjected to the same exact extraction and qPCR procedures as the test sample. The inclusion of a negative control sample can help to demonstrate lack of systematic cross-contamination and therefore prevent the false positive result. Another important control within the whole cell DNA method is the positive control. Typically, for a positive control, one would spike in a known quantity of DNA into the test sample to demonstrate satisfactory DNA extraction and qPCR amplification. This is a in-run control, and the good practice prior to any new matrix coming in is to actually perform a more thorough initial interference study to make sure that a matrix does not have any severe effect on the extraction as well as qPCR amplification efficiency. Importantly, as I've just suggested, all these controls should be subjected to the same treatment as a sample and included in each assay run to ensure the assay validity. Other system suitability criteria that are typically implemented in whole cell DNA qPCR methods are the linearity of the standard curve. Typically, a 0.98 criteria is applied for the standard curve. And one would also want to look at the PCR amplification efficiency. And for that, the slope of the standard curve is usually assessed, and a typical range is between -3.0 to -3.8. The accuracy and the precision of the method has to be validated for the particular method, and this can be done using a replicate sample and sample spikes with known quantity of DNA at multiple levels, and definitely include one that's near or at the detection limit or quantitation limit. The limit of quantitation obviously has to be sufficiently low to detect DNA level at or below specifications. Now, for whole cell protein, it's pretty common that ELISA method has been used predominantly. There are different commercially available, or what we call a generic assay kits, available for different host cells. These are ready-to-use reagents and often include both the whole cell protein standard as well as the detection and capture antibody. These are a very convenient choice during early phase of product development, especially when a sponsor has not determined the final purification steps of their product. Again, the commercially available assay kit has been used pretty commonly during the early phase of the project. However, as the project moves into a later phase, a process-specific ELISA oftentimes are developed for a specific production strain and manufacturing process. And the whole cell protein generated through the sham production run. Basically, one would subject a whole cell line without the product insertion to the same upstream as well as downstream process and produce a whole cell protein to be used as a reference standard as well as the immunogen. If a company have multiple products that use very similar or comparable processes, a platform method may be developed that can be used for multiple products. And as me and Sheila has specified, the process-specific ELISA are often preferred and expected by regulatory agents for late-phase product unless one could assess and confirm the suitability of a commercial kit. Now, for whole cell protein, a method is really as good as its reagent. First of all, generation of the whole cell protein standard, which is also the immunogen and the antibody, is a crucial step. Not every single protein in the whole cell population is immunogenic. So appropriate whole cell protein adjuvant and optimized immunization procedures need to be implemented to make sure the whole cell protein prep represents as wide as possible the population actually seen in the process. The antibody needs to be purified to ensure affinity as well as stability. There are different ways of protein purification, such as protein A column or A or G column, or one could apply a whole cell protein affinity column. Both have its advantages and disadvantages. For example, the whole cell protein affinity column can provide a highly specific and pure antibody. However, if the whole cell protein bound to the column is not quite as representative, then one could potentially lose a certain antibody population and therefore lose the reactivity to the whole cell protein. The antibody also has to be conjugated properly for the detecting step of the ELISA. Common conjugates are biotin or HRP conjugates. The other very important thing to consider is the characterization of the whole cell protein antibody. Whole cell protein antibody generated should have sufficient reactivity towards as many whole cell proteins as possible within the population. A standard way to look at that is to use a Western blot using the whole cell protein antibody and compare that to a stained 2D gel and see how much of the protein is represented and detected by the whole cell protein antibody. However, this typically is a qualitative measurement, and percent coverage is more of an arbitrary and should be used with careful considerations. Another method to characterize the whole cell protein antibody is actually to do a use test. In the use test, one would use a typical production sample and most likely from various steps of the purification steps to compare the new lot of reagents versus the old lot, or to compare the process-specific antibody versus the commercial reagents to demonstrate that the ELISA methods and the whole cell protein antibody can pick up the various populations of whole cell protein within the purification steps. One important thing to consider for whole cell protein methods is dilution linearity. Whole cell protein methods validation poses unique challenges. This is because the whole cell proteins and whole cell protein antibodies are both mixed populations. So the whole cell protein composition present in a given test sample may be significantly different from the whole cell protein standard and biased towards a certain protein population. So for this reason, sometimes one can observe lack of dilution linearity in certain sample types, and this was potentially caused by insufficient antibody for some whole cell proteins in the population. One needs to remember that satisfactory spike recovery does not always equate to dilution linearity. I think almost everybody understands this spike recovery study is needed. However, sometimes the dilution linearity study was ignored because satisfactory spike recovery was obtained. So for best practice, for samples with whole cell protein levels that are below limit of quantitation, one should demonstrate satisfactory spike recovery at multiple levels, again including one level at or near the limit of quantitation to make sure that the matrix effect does not impact the limit of quantitation. For samples that have measurable amount of whole cell proteins, and these are typically in-process samples, one should demonstrate dilution linearity by testing at multiple dilutions, and the sample values within the linear portion of the validated range are averaged based on pre-established criteria, and oftentimes a CV percentage of no more than 20 or 25% is often applied. So I want to leave my presentation with just this slide. This is a summary of some of the USP initiatives on whole cell protein and whole cell DNA measurement in biopharmaceutical products. For the residual DNA testing, the new Chapter 30 is being drafted. It will provide extraction and qPCR procedures, and it will also provide primer and probe sequences for CHO and E. coli methods, given these are the two most commonly used methods within the industry. The DNA reference standard for CHO and E. coli methods will also be created, and the USP committee is also revising Chapter 1130, specifically on the residual DNA testing, and the draft will be available soon later this the draft is actually available right now. For the residual whole cell protein, the USP panel is writing a new chapter, 1132, and the draft will be open for comment later this summer. And again, thank you very much for attending our webinar. Rita, I think it's back to you. Thank you.
Yes, thank you very much, Dr. Wang. I'd like to thank all the presenters for their insight and information. At this time, we'll begin the Q&A portion of our program. First, I'd like to remind the audience on how to submit questions. You can submit your inquiry by typing it in the Q&A box, which can be found by clicking on the red Q&A widget at the bottom of the presentation window. We'll try to answer as many questions as we can. So here's our first question, and this one is for Dan. What determines which detector types would be used if several different system types would be effective?
Well, I think the most important thing to consider is what your detection limit you need to achieve will be. If you can live with a higher limit of detection in the parts per million range, it may be ideal to stay with a more traditional detector like charged aerosol or refractive index, and then decide if your levels have to drop after that. You can move on to a more advanced technique. But it really all depends on which or how low your detection limit needs to be, and that's a decision made with the manufacturing team and the safety group and so on.
All right, thank you, Dan. Sheila, this one's for you. How critical is it to identify all impurities in the drug substance?
I hate to give you the FDA answer, which of course is it depends. It is important to measure those which are present or are shown to be present or created during degradation. You want to understand what the degradants are. If levels are very, very low, it is often acceptable not to be able to identify them, but it is important to be able to identify those which are product-related, as those may have biological impact on the drug itself.
Okay, thank you. It's a question for Wei. For host cell DNA assays, what is the acceptable sample spike recovery?
I think for the most common acceptable limit is about 50%, so 150%. I think this is the proposed acceptable limit within the USP expert panel. However, everybody recognized that each matrix is very different, and for certain matrices that are kind of difficult to achieve recovery at that level, certain correction factors may be used, but that should be used with careful consideration.
Okay, Dan, back to you. If multiple separation systems will work for a residual target, what is the decision process that goes into the choice of column chemistry to be used?
What we would normally look at if multiple separations are possible is what is going to work best with our sample preparation. So in other words, is it a technique that is going to allow for a very smooth transition of injecting the sample onto the system without doing buffer exchanges, without doing a lot of manipulation, as well as does the column need any special care and consideration if it does in fact come in contact with protein, which isn't necessarily a terrible thing, but can shorten the life and cause more cleanup than necessary. So it really depends on what type of sample you're dealing with and what is going to be in the material that's injected to really make a decision on the column chemistry, because you may have some unintended retention of components of the sample if the column chemistry is not chosen carefully.
Okay, thank you. Sheila, can impurities that are below the level of quantitation be left out of purity calculations?
This is something where it would be better to include them. It is fine to include them as a sum. That is, if you have 10 peaks in a chromatographic separation that are clearly impurities and all are below the level of quantitation, it is acceptable to note that there are 10 peaks below the level of quantitation but above the level of detection. That gives a more accurate representation of the purity of the material. If one just leaves them out, it will make the material look as though it is purer than it truly is.
Okay, thank you. Wei, question for you. Is there a distinction between residual host DNA and plasmidic DNA?
I don't think there is a specific regulatory statement distinguishing the host cell DNA versus the plasmid DNA. I assume most of the plasmid DNA contamination comes from, for example, the E. coli fermentations, etc. I think we should treat them similarly to the host cell DNA with respect to the acceptable limit in general.
All right, here's another question for you. What % of HCP should the HCP antibody recognize?
This is actually a difficult question. I think people like to see a high percentage of the host cell protein being recognized by the antibody. However, keep in mind the 2D Western and stained gel comparisons is really a qualitative method. And for that reason, actually, there's a recent host cell protein presentation by USP. The expert panel working on the USP host cell protein chapter has advised against a percentage or the quantitative criteria for this assessment. So I think if one can do a qualitative assessment to make sure, for the most part, all the protein populations in the four quadrant of the blood is relatively equal represented, and plus use a use test to provide some quantitative results, that will be a better practice rather than focus on how many percentage of the protein is covered by the antibody, because that number is really objective and cannot be stand up to further investigations.
Okay, Dan, I have a question for you. How do you measure impurities that may be bound up or retained as a reservoir associated with the API?
That's a good question, and we have observed this with certain impurities that may be sequestered. And really what you have to do is look at the potential use of sample additives. And we've looked at that with a couple different ways. One thing you want to try to do is look at increasing and decreasing concentrations of sample additives, items like tritons or other surfactants that could potentially allow for the release of materials that you're actually trying to identify. You're going to have to look at many different concentrations, and you're going to have to look at differences with and without to actually get a feeling if your target of choice is being hung up on the actual API.
Okay, thank you. Wei, a question for you. If you perform HCP ELISA at different dilutions, which final result would you use to average?
First, the uncorrected results have to fall within the linear portion of the method itself. So with that, you also want to look at the overall variability between the corrected sample results. And typically, as I suggested in the slide, a criteria of no more than 20 or 25% of variability should be observed for all the results that are averaged.
Okay, we've got a few more questions here. Wei, this is probably for you. When will the new DNA Chapter 30 be available for review, and where?
The draft Chapter 30 is being reviewed internally within USP. I suspect it will be available later this year because there's still a lot of validation work associated with it. I'm not 100% sure where it's going to be available, but within the USP website, I'm sure you can find the information.
Okay. Let's see what else we have coming in questions here. Hold on. Sheila, are chemical residuals such as antifoams, small molecules, et cetera, typically put on product specification and transferred to a QC lab?
Early in development, it is very common to have that done because the clearance studies have not yet been completed. One would also do a risk assessment. In some cases, it is clear from the risk assessment that even if antifoam, for example, were still present in the product, it is below the level of detection of the commonly used methods. In those cases, it wouldn't be included, but the risk assessment would have to be part of the filing so that the agencies would understand that this has been done and it hasn't been left out. For protein residuals, such as insulin or benzonase, in general, it is expected that early in the process they will be on the specification. Those methods will be running QC on the final drug substance. But as development proceeds, the clearance study should demonstrate that the material is removed, and then they will be removed from the specifications.
Okay. We have time for one last question. Sheila, this is for you also. If DNA is inactivated, will this allow for acceptance of higher residual levels by regulations?
This is one of those slippery slope questions. If it is inactivated, you have to prove that it's inactivated. Again, proving a negative is often difficult. Additionally, for many regulators, it becomes one of those accepted process that they just want to see this level low, even if it's inactivated. I do know of situations where individual companies have demonstrated that while the DNA is present, it has been chopped up into small enough pieces that it could not be hazardous to the patient. In those cases, a higher level was allowed.
All right. Well, thank you all for those insightful answers. Unfortunately, we're out of time. If your question was not answered during this presentation, it will be addressed via email. I want to thank the audience for their interesting questions, our speakers for their presentations, and our sponsor, Eurofins Lancaster Laboratories, for making today's education webcast a valuable experience. This webcast will be available for on-demand viewing through April 2015. You'll receive an email from Pharmaceutical Technology alerting you when this webcast will be available for replay. Please invite your colleagues who may have missed this live event by forwarding them the announcement. Thank you again for attending today's webcast, Navigating the Analytical Development Challenges for Bioprocess Residuals and Impurities. Thank you once again.